enzyme kinetics, the rate at which the enzyme work is called enzyme kinetics. Michaelis and Minton said that enzyme combine with substrate to form enzyme substrate complex, and they frequently break down to form enzyme and product. And these reactions were assumed to be a reversible reaction. E plus S forms ES, that is reversible, and again ES forms E plus P, means product. Enzyme kinetics is a function of concentration of substrate available to enzyme. We have plotted the graph to study kinetics. Substrate concentration is plotted at x and enzyme velocity is plotted at y-axis, and hyperbolic curve is obtained. M and O are the stages of reactions when the substrate concentration is very low, and it represented as M, and when the enzyme velocity reaches maximum, it is represented by O. When the activity is half of the maximum, it is represented by N, and the rate of reaction is directly proportional to substrate concentration. Michaelis-Menten reaction is V is equal to Vmax multiplied by S divided by Km plus S, where V is equal to the velocity, Vmax is maximum velocity, Km is Michaelis-Menten constant expressed at moles per liter. Km represents the substrate concentrate at which the velocity of the reaction is half of Vmax, and Km is inverse measure of affinity and strength binding between enzyme and the substrate. The lower the Km, the greater is the affinity. Point M, at this stage the substrate concentration is less than Km, and if the substrate concentrate increases, its value does not change, and S can be ignored at denominator. V is equal to Vmax upon Km. Point N, at this point the substrate concentration is equal to Michaelis-Menten constant Km, so the equation becomes V is equal to Vmax multiplied by S divided by S plus S, and V is equal to Vmax multiplied by S divided by 2S. V is equal to Vmax upon 2. Point O, at this point the substrate concentration is higher than in comparison to Km, so that Km may be ignored. So V is equal to Vmax multiplied by S upon S. V is equal to Vmax. So Lean-Weaver-Burke equation is to plot the data conveniently. Michaelis-Menten equation is transformed to Lean-Weaver equation. 1 upon V is equal to Km plus S upon Vmax multiplied by S is equal to 1 upon V is equal to Km upon Vmax multiplied by S plus S upon Vmax S is equal to 1 upon V is equal to Km upon Vmax multiplied by 1 upon S plus 1 upon Vmax. This is a Lean-Weaver-Burke equation, and this is a plot. I hope you can understand that. Km is not a fixed value and vary with structure of substrate, pH and temperature, and factors affecting enzyme activity are concentration of enzyme, concentration of substrate, temperature, pH, activities and oxidation. Concentration of enzyme. The increase in concentration will increase the catalytic site because more catalytic sites are available to perform the reactions. So more the concentration, more the catalytic sites, and more useful will be the reactions. Another one is concentration of substrate. When the substrate is low in quantity, the rate of enzyme is low because of the all catalytic sites are not occupied by a substrate molecule. So when the substrate will increase, it will automatically increase the activity of enzyme, and it reaches a maximum point that becomes constant. Third is temperature. The rise in temperature accelerates the rate of enzyme catalyzed reaction up to certain temperature called optimum temperature, and if the temperature is higher than the maximum temperature, the enzyme become denatured. For a human beings it is 37 degrees, and for the plants it is 60 degrees. So you can see the plot, and A is optimum temperature. After that the enzyme become denatured. pH. Every enzyme has optimum pH, like optimum temperature. The pH activity is due to the ionic nature. Within the narrow pH change, the changes are reversible, but if pH is too high or too low, it becomes denatured. Most of the enzyme ranges between 5 to 8. Pepsin and trypsin are active in highly acidic medium 1.5 and highly alkaline medium 8. Activator. Activators are the certain ions that influence enzyme activity. Enzymes such as hexokinase requires ATP as need Mg plus 2 magnesium ions. Without these ions, they become inactive. Oxidation. Some enzymes having sulfur-hydryl group are sensitive to oxidation due to the oxidation of SH group by aryl oxygen. A disulfide linkage is formed with a loss of enzyme activity that can be further restored by the reduction of enzyme by cysteine and glutathione, etc. Enzyme inhibition. What is enzyme inhibition? It is the inhibition of enzyme action. So there are three major classes of enzyme inhibition. One is competitive, non-competitive and uncompetitive. Competitive means there is a competition between two substrate and inhibitor. The inhibition takes place when a compound having strong structural resemblance with substrate compete for the catalytic site. One of the compound bind to the site, an enzyme cannot complete its action, but if the concentration of substrate is increased, it will again activate the activity and product will be formed. So this is reversible in nature. Example, succinate dehydrogenase is enzyme in the citric acid cycle where succinate gets converted into fumarate. So succinate is a substrate, fumarate is a product, and compound malonate, that is an inhibitor, and if added to the reaction it reduces the product formation. So here malonate and succinate both are having strong structural resemblance. So on the catalytic site of succinate dehydrogenase, so malonate, if malonate get attached to the site earlier, then succinate cannot convert properly into fumarate. So if we add more succinate, then the reaction further starts again and make fumarate. So non-competitive inhibition, the inhibitor binds to the site other than the catalytic site, hence there is no competition between the two. It cannot be reversed. Enzymes with SH group are inhibited by the metals like mercury and copper. And uncompetitive, here the inhibitor binds with complex, the enzyme substrate complex, and makes it inactive. Due to this, it is very difficult to make products, but this kind of situations are very, very rare.